in a very real sense, what is breeding value? And one of the things which, as I say, additive genetic variance has a formal definition, which is the covariance between the sum of the covariances between average effects and average excesses. I'm not giving you that definition other than quickly and verbally, for the simple reason that it's useless. It's not something that can be measured. And there's lots of reasons why it has real issues. And so what we can do is, the nice thing is it has corollaries. And one of the corollaries is that additive genetic variance is the variance of breeding value. The breeding value has this definition, which is twice the deviation of your offspring of an individual's offspring from the population mean. What does that mean? It means that what we do is we take a sire, in most cases if we're dealing with animals, we'll take a sire, mate it to a large number of dams of females, and we'll measure their offspring for the trait we're interested in, right? And so we can get a breeding value for everything. For example, we can measure bulls, and if you have a star bull, it will be mated to a large number of cows. And every time you do that, you write down what the mean phenotype of the offspring is in the stud book. So that's what a stud book is. It is a book of what the offspring of a particular sire or dam looks like, what their phenotype is. And the average of all that becomes a breeding value. So we know the breeding value of star bulls very, very well. And this is actually one of the statistical complications we have to deal with, is that statistically you should use random mating and everybody should have an equal chance. But when we actually deal with real populations, you aren't going to breed your bad bulls. You're only going to breed your good ones. And so we have to use fairly sophisticated statistics to do that. But what that means is, for example, a trait might be milk yield. And so we would say, for any given bull, we might mate that bull to a cow and take her female offspring and measure the milk yield of that cow. And that bull now has a breeding value for milk yield. And so a quantitative geneticist will ask with a straight face, what is the milk yield of that bull? And they can do that because they're talking about the breeding value. It's how many gallons does the offspring, the female offspring of that bull produce. Now there's a couple other things. Say you have Charolais cows and bulls, and you get a breeding value for that bull in a Charolais herd. And now you want to move it over to Herefords, another breed. The breeding value changes. So what you knew about how it worked in the presence of Charolais may not tell you how it works when it's mated with Herefords. And so again, it's a the breeding value is a function of the genetics of the individual in the context of their population. And that's something to be very aware of. Now, where else might we use this is that one of the things which a breeder will do is they will set out and try to improve something. Say they want to produce a cow that has high milk yield and high butter fat. And they'll create an index, which is essentially economic value. How many a cow that produces lots of high butter fat milk has a high economic value. One that has low milk or low butter fat will have a low economic value. And what they'll do is they'll do a breeding experiment. And or maybe they already have it in the stud books. And they'll ask, what is the heritability of this economic index that they created? What is the heritability of economic value? That's the phenotype, economic value. And if the heritability is fairly high, they'll say, this looks promising, and they'll continue on and do the breeding design. On the other hand, they may do the selection experiment and try to get higher yield. On the other hand, they may discover that the heritability is very low. And at that point, they'll say, it's not worth it. It's not economically viable to select for this trait because it's not going to work. Additionally, the other thing I want to get that I think is really important is that we think a whole lot about molecular biology. One of the interesting things is that, you know, when you get good bacon these days, when you get nice big eggs, those are the product of quantitative genetics. The thing that is really wonderful about quantitative genetics is it works, it pays the bills. And it is the kind of genetics that actually works. Now, I was also quizzed about, yeah, in a real sense, it pays for itself. I was also quizzed about epistasis a little bit more. And the question is, how would we actually deal with epistasis in terms of average effects? And what I thought you should think about, you know, what are these things? Everything is a deviation. The mean of all of those epistatic interactions is 0, because that's by definition. And you can say, why are they independent from each other? Well, if you're familiar with regression, what we're doing is just a gigantic multiple regression. And if people want to see this, if we have time, I'll show it to you. But we're doing this gigantic multiple regression. And what we're doing then is, because it's a multiple regression, if you remember from your simple regression techniques, when we do a regression of y on x, we divide any given value of y into that part which is due to x, which can be predicted by x, and a residual. And the multiple regression, what regression does is it divides those into 2 orthogonal independent components, so that the predicted and the residual is always independent of each other. Same thing happens with multiple regression. All of those values will always be independent of each other. But just as I showed, you know, if we go to this graph, you know, if we go to this graph right here, yes, they're independent of each other, but yes, they're independent of each other, but when we change gene frequencies, when we change the, which is effectively changing the weighting on those 3 points, the regression slopes and lines change. So yes, that's the beauty of regression, is it divides everything into independent components. And the horror of regression is that it's a function of the genotype and the population. Now see, these things don't actually change, right? The blue dots don't actually change. They are fixed quantities. And what I am doing in this is I am doing a regression on these 3 points, on the number of A1 alleles, 0, 1, or 2 A1 alleles. And so what that's doing is, these 3 numbers, it's always only those 3 numbers, but they get added with different weights. And we do our regression, and it changes the slopes. Again, it's something you can do with Excel pretty easily. And so that's how those change. And the same thing is going to happen when we do this multiple regression with with dominance and epistasis and whatever else you want to throw into the equation. That we're going to get that same sort of fitting the best plane through there. Now, that raises the last interesting question. How do we relate? We can say, what is the additive genetic variance for the A locus if it's interacting with the B locus? And so that implies that we're going to get some sort of a single bivariate line like this when we look at the A locus. However, we know that it's this plane. So what is that additive genetic variance for the A locus? Well, it turns out what it is the projection of the B locus onto the A locus. And again, it's a weighted projection. And so if you could imagine that well, it's probably easier to project onto the B locus to get an idea. But for any given gene frequency, that plane changes. And we can imagine that those planes were more or less transparent. And the higher the frequency of that genotype, the less transparent it is. And if we were then look down this way, look down the edge and just sort of look at the shadowing on a shadow box. So that's what a projection is. So we're projecting just on a bivariate axis right here. And what we're doing is, some of the lines are sloped, some of them are flat, And if you went over here, they'd be sloped the other way. And all of those genotypes, all those B locus genotypes exist, all those A locus genotypes exist. Well, but they have different weights. And if we were to think of those as being, those weights giving them different levels of transparency, then we were to look down that projection, we'd see a cloud. And we could sort of draw a line through where that cloud was densest. And that would be our projection. So in fact, it ends up being absolutely no different. What did I do? It's not what I want. Oh, it's going the wrong direction. Oh, boy, I'm sorry. Okay, I was trying to get here. Okay, I keep pushing the wrong button. Okay, this is what I was looking at. Fisher was dealing only with a single population of very large size. A lot of very funny things happen with his, what's called his infinitesimal model. But in fact, this cloud here is not just environmental variance, it's also epistasis. And so it really is no problem to focus on one allele, as long as you're willing to admit that when gene frequencies change in another locus, you're going to have to redo everything. Okay, any other questions on this? Should we go on and think about making it even more complicated? Okay, so multiple traits. Well, this is what we know up to this point, right? What I showed you at the top. And it turns out that we can actually move to multiple traits as simply as moving to linear algebra. So that P, instead of being a single phenotype such as milk yield, becomes multiple phenotypes that are, or a multi-dimensional phenotype, which are arrayed as a vector. And so we could have milk yield, gallons of milk, butter fat, disease resistance, something like, I don't know, sugar content, I don't know what else you have in milk. But you can have as many as we wanted. In theory, we could have a very large number. I will say that in practice, if you go over about 4 traits, you'll hate yourself. Anyway, just as we divided that into additive, dominance, epistatic, additive by dominance, dominance by additive, dominance by dominance, etc., we can do exactly the same thing. We can look at a large number of traits, we can divide into a set of additive components, a set of dominance, a dominance vector, an epistatic vector, etc. And that's all there is to it. We're done. We can go home. Okay, the problem we have is that this is, the problem we have with that, of course, is that we can't just look at an individual. We need to move to a variance. And the way we do that is simply squaring those individual vectors, and that gives us a covariance matrix. And so, in fact, this VP becomes this, which has, along this diagonal here, what we have are the phenotypic variances for each of the individual traits. So again, milk yield, this is a phenotypic variance in milk yield, this is a phenotypic variance in butter fat, phenotypic variance in disease resistance, what have you. Along the off diagonal, what we have is the covariance. So the reason I said milk yield and butter fat is that there's a negative correlation between those. If you increase milk yield, the easiest way to do that is to increase the volume of milk. And as a consequence, the butter fat goes down. I don't really know what the covariance is for cows. This is Brazil and everybody eats beef here, so that's why I'm using that. But I do know that for chickens, egg size and egg number are negatively correlated with each other. It's very hard to create a chicken that produces lots of large eggs. You get lots of medium eggs, and a few extra large eggs. Really hard to get a lot of extra large eggs. And that's because they have a negative covariance between them. And that's described by this term right here. So that's really what that's telling us right here. So this is a phenotypic covariance matrix. This is the additive genetic covariance matrix, that term right there. And at this point I got really bored, and I decided not to write that out for everything else. Now, how would we get, how do we know how to get VP, right? VP 1 through N and all that. We take all of our cows and we measure every one of them for every one of the traits. And so for each individual, we have this vector of phenotypes, and we can then use that to calculate what the phenotypic variance is. Well, to get the additive genetic variance, we're going to do exactly what we would have done before. We're going to do a regression of offspring on parents, for example, or we could do an analysis of, or nested analysis of variance, but and we could get each of those variances. And what that would do is, of course, that would give us a separate variance for each one. But how do we get these covariances? Well, essentially, linear algebra does it for us. If we do a man over it, we'll just simply kick it out. Or if we do a multiple regression of this sort, we'll just simply kick it out of the system. But basically what we are doing is we're doing we're saying what is the covariance between, we're saying what is the cross product. So with variance, we would take milk yield and square it, and sum up that squared milk yield, because remember, deviation is 0. For the covariance, we would take milk yield times butterfat, and we would sum that up, and we would get the covariance. Now notice the difference. Variance is always positive. Covariance can be 0, it can be negative, or it can be positive. How about this one? Well, what we need is exactly the same thing. We want to get the covariance between the offspring in exactly the same way. And what we would generally do for these, it actually turns out if we did a regression, is we would do a regression of offspring butterfat on parental milk yield, and vice versa. And they should give us the same number. So anyway, if you have SAS, you don't need to know that. But in practice, you can in fact do it by hand. You would really hate yourself. So we can now deal with the multiple traits in exactly the same thing. Here's our breeder's equation. And what we have is, again, we have S, which is the change in the mean phenotype as a result of selection. By the way, when I was describing it last time, I said Z was the population before selection. This is the population that results from those that are allowed to breed. But we could be more subtle than that, right? Z bar star could be the population being weighted by the number of offspring we have. So that individuals that produce lots of offspring would have lots of weight, and ones that produce only one offspring would have a little bit of weight. So this is just really the weighted sum, and that's the unweighted sum. And this is weighted by relative fitness. This is the mean of the difference between the mean of the surviving of the offspring and the mean of the parental generation. And this is a ratio, constant proportionality, that converts additive genetic variance, change within generations into change between generations. And as I say, in my mind, I don't know how to do this, but in theory, this could be a much more complicated. This should be f of p, a function of phenotype, which could be arbitrarily complex. So anyway, this part's easy. It turns out that S is a vector. Okay, so for every given trait, we can take what is the mean of the population. So the mean of the population after selection minus the mean of the population before selection. And so we just simply take all those individuals that are allowed to breed or weighted by the number of offspring they produce, take that mean and compare it to the unweighted mean. And we do that for every trait, no matter how many there are. Similarly, we can do that for the offspring. And by the way, these are two empirical things, right? We can measure, we can do the selection, we can measure S, we can look at the offspring, and we can measure Z. What are we going to do about this? Because if you know anything about linear algebra, it actually turns out that you can't divide. And so what we have to do is, we actually have to divide that into a pair of matrices, where this is the additive genetic variance covariance matrix. I'm sorry, this is the phenotypic covariance matrix, but it's been inverted. And again, inversion is an ugly thing to do, but Mathematica will do it for you. I had to program it once. It was not fun. And, Jim, that's actually why I was talking about adding linkage disequilibrium to genetic drift, and the reason I had issues with that is because I had to invert these gigantic matrices. Anyway, this is the full move to linear algebra. And so here's R H squared S. And again, we know what these are. This is just simply the inverse of the covariance matrix. So we can do exactly the same thing. There's no magic here at all. Now, what we find is that often that's not the convenient way to work. Because often we take P inverse S, and we call that the selection gradient. Now, why would that be? Well, think about this. Say I select on milk yield. And milk yield and butterfat have a negative covariance, as we might expect, because, as I say, milk yield is easiest done by just adding water to the milk. In that case, if I select only on milk yield, butterfat is also going to change. So this is just the raw mean of the population before and after selection. And so even though we're only selecting on milk yield, these other traits happen as well. And this is what's called indirect selection. So when we're selecting for milk volume, we're going to select for, we're also going to indirectly be selecting for, or affecting, butterfat. However, we would rather know exactly where selection is acting. And it turns out by magic, which isn't always very obvious, P inverse S, that gradient, shows you exactly where selection is acting. It removes that correlated, that indirect selection from the equation. And so multiplying these two together gives us this gradient, which is very useful in saying where is selection acting. Now, of course, if we're breeding cows, we know where selection is acting because we are the breeders. And we're the ones who go out and say, I'm taking you because you produce lots of milk. On the other hand, if we're out in nature and we see that, and we're studying something in nature and we discover that the lizards have gotten longer and they've also gotten fatter, we might want to know whether it was selection for body length or selection for body weight. And in fact, the classic question that gave rise to this was by Russ Landy, and he was asking the question about brain-body size allometry. What you find is that small animals have small brains, and big animals have big brains. Now, is selection on body size, brains, or both? You know, as you might expect, there's a positive correlation. The gradient actually is going to tell us that, because it's going to remove these indirect selection limits. It's also rather nice because it gives us this G matrix, which I'll never forgive Landy for. I should have called it the A matrix. And he said you have this A matrix, which is G matrix, sorry, which is the actual genetics of the system. So I really like that. We're dividing this into phenotype and genetics. And I should point out also that if you're actually designing experiments, there's really two parts to most experiments that you're going to do about evolution in natural populations. Oftentimes you'll go out into the field and you'll, a standard experiment that was done by Steve Arnold was he took a bunch of newborn snakes and he measured them for their sprint speed. And then he released those, and he tagged them all, and then he released those in nature. And he came back a year later, and he captured as many snakes as he could, and recorded their marks. Well, that gave him S. That gave him the selection differential. He had the recording of all of the snakes, given the sprint speed for all of the snakes, and he had the sprint speed of those snakes that survived. Plus, although he may not have caught them all, but if you assume that that's a random sample, that's okay. And so he had S. And how did he do that? The important point about that is he went out in nature. If you're going to look at selection in nature, you have to look at selection in nature. You can't do an experiment in the laboratory to see who's going to survive when you release them out in the wild. On the other hand, genetics is a very different thing. He then set out to figure out what the genetics of the system was. And what he did was he bred the animals in the laboratory. And he measured the sprint speed of offspring that were related to each other, and he was able to calculate the additive genetic variance from the proportion, by using relatives. In other words, this part has to be done in nature, and this part is often done in the laboratory. And so that's the kind of thing that we have that I think is another very useful thing about quantitative genetics, because what it does is it allows you to recognize that there's a difference between selection, which is the ecological process by which some individuals reproduce and others don't, and genetics, which is the transition equation which converts the selection differential into change in the next generation. So anyway, so we have this, this gradient, we have the G matrix, and we have the response to selection, which is, again, these are two empirical things that can be directly measured. Okay. What this gradient is it is the vector of partial regressions. Vector of partial regressions of relative fitness on phenotype. So in this case, phenotype 1 holding phenotype 2 through n constant, phenotype n through. So in other words, we're going to do a partial regression of relative fitness on milk yield holding butterfat constant, if you will. And so this is kind of an interesting thing here. Because what that does is it points directly uphill. It always points uphill. And by doing that, that's confusing. Okay, so here it is. Here's our optimum. This is the idea of an adaptive topography. What an adaptive topography is, this was introduced by Sewell Wright. Here is trait 1, milk yield. Here's trait 2, butterfat content. And here's our optimum. And so this is what we're aiming for. This is our best phenotype, our target phenotype. And then this is what those dark lines are, the black lines are, is essentially topographic lines. So this is uphill. Okay, so this is low fitness, this is high fitness, and you can sort of see that going from light yellow to dark yellow. Okay, so there's our optimum. This is where the population is. And what beta does is it always is going to point directly uphill. It's going to point exactly at this optimum. Okay? Regardless of where the population is on this, it always points directly uphill. And of course, the S vector isn't going to do that because it's got the correlations in it. But the beta vector actually has removed, by putting these two together, we've been able to remove those indirect selection, and we're going to actually point directly uphill. Okay, so this is where the G matrix comes in. And the G matrix, one of the things that you can do with linear algebra is you can think of a matrix as matrices are sometimes called rotation matrices. And what they do is they take a vector and they move it into a different space in a different length. And they move it in some sort of random, in a particular way. for a particular matrix. Well, the thing to notice is that that beta matrix is a vector, and this is the standard way of describing what a vector is. It's moving straight uphill. It's an arrow that points straight uphill. And then what the G matrix is going to do is it's going to rotate that. Now, if the correlation is 0, what that means is that you have the additive genetic variances, but they are unrelated to each other. So if your arrow points directly uphill, the population is going to go straight uphill because it doesn't rotate the response vector. On the other hand, if the correlation is something other than 0, and here I said it was positive, what you do is this circle becomes an ellipse. And that ellipse may not be square to the original traits you've described. By the way, I drew this as a circle, but it doesn't actually have to be a circle. We could expand, if we could imagine that VA1, if the additive genetic variance for the first trait was big, this could be out here, and this could be narrow. It would be an ellipse. But the ellipse would have its major axis and minor axis exactly parallel to the A and the B axis. What's so special when the covariance is greater or less than 0, when it's not 0, is that the axis does not line up with the original traits. And for those of you who are into statistics, you may know about principal components. And principal components is a transformation. So if you start out with 2 traits, and you do a principal components analysis, you end up with 2 traits. Well, what those 2 traits are is a linear combination of these 2 traits that gives you this axis and that axis. So in other words, what it's really kind of saying is that the traits you measured were the wrong ones. The traits you should measure are the principal components because they're independent of each other. And so that's what the G matrix is going to do. It's going to take this axis and it's going to rotate it into a new set of traits which evolve independently, if you will. But of course, we're selecting on only one of them. We're not selecting on that index. So, here's our optimum. Okay. And here is our population. And I've drawn our population differently now because I've said, rather than just putting a point, what I've done is I've put a distribution. And this distribution is now circular. It's essentially the 2 traits have the same genetic variance and have a covariance of 0. And so this population is right here. The arrow points exactly uphill. The gradient points exactly uphill. And this population will climb straight up to the optimum. And it's not a problem. We can create whatever index we want. We can select for milk yield and butter fat. We're going to go straight there because it's really no problem to go along this line. But look what happens when when there's a genetic correlation. The gradient still points right here. I should have put the gradient in. The gradient points straight uphill. But it's a lot easier to evolve along this axis than it is along this axis. And so it will move much more rapidly along that direction. That's the rotation we're talking about. And so this population will move along a very long arc to eventually get towards that. So this sort of draws in another perspective. Here's trait A, here's trait B. We're going to select straight uphill. We're going to select only on trait A, but trait B is going to go along for the ride. And it's because although the gradient pulls that way, it's easier to move along this direction. And it will actually move along that axis to the extent that it's capable of. And so this is your correlated response to selection. So it means that if we select on one trait, we're going to change the other one along with it. So here's our adaptive peak, here's our optimum, here's our topography that I'm talking about. And we can put our population wherever we want. And so here's beta, which is pointing straight uphill. And here is our response vector, which is going to be not straight. It's not going to, it's not going to be parallel to that. It's going to go along, it's going to be rotated by the G matrix. It's going along the direction of least resistance. Now this is a really interesting thing, because if we look in multivariate space, what we would do is, in this case, two-variant space, we start out this far from the top, the optimum, and we end up this far from the optimum. So in fact, we have moved towards the optimum. The response has been positive. However, if we were to only look at trait 1 here, trait 1 is pretty close to the optimum when we start, and we select by selecting on trait 2, and trait 1 moves away from the optimum. You get a very counterintuitive result. The population was at the optimum, and when a selection is applied, it moves away from the optimum. Well, it turns out it doesn't move away from the optimum in multivariate space. But when we look at a single projection, it appears that, in fact, it has moved away from the optimum. So if we now look at this, the population will spiral in to the optimum. And what's interesting, if we look at trait 1, it gets worse, then it gets better, it overshoots the optimum, and then it overshoots the other way. And so what it does is it does a wobble, and eventually it will get to that optimum, but it may be a very, very slow process. Okay, so that's really, in essence, this is really all I wanted to say about multivariate evolution, multivariate selection at this point. And we can move, but we can actually talk about, I mean, I have a few other slides, but that's really the core of what I wanted to talk about. Now, the question is, what causes genetic correlations? Because what I've told you about here is, I've said there's genetic correlations. I've talked about these genetic correlations as if they were sort of esoteric things. Egg size and egg volume, they're negatively correlated. Milk yield and butterfat are negatively correlated. Now, what could cause a genetic correlation? Well, I think you can see the hint. That is, if you have a gene which is going to increase the volume of water in the milk, it's going to lower the butterfat. And that's really called, that's pleiotropy. Basically, pleiotropy is when one gene affects multiple traits. So, for example, gene C affects traits 2, 3, and 4 in this example. And so you have one gene affecting multiple traits. That's really what causes genetic interactions. There's another thing which can cause it, which is linkage disequilibrium. But that only becomes important in very special circumstances. The only one I can think of that's really common is in sexual selection, which we will not be talking about. But for the most part, what causes pleiotropy is one gene affecting multiple traits. And sometimes it's really obvious. As I say, milk yield and butterfat content. Well, if you're just adding water, a gene that causes adds water to the milk is going to increase volume, but you're going to be diluting the same butterfat further down. Here's an example with sickle cell anemia. It is, of course, a very nice example. You can't read it because I don't know why. But basically what it's talking about is that, of course, we know that sickle cell anemia is a single amino acid replacement, which is due to a single SNP substitution, single nucleotide polymorphism. However, it turns out that the sickle cell heterozygotes have a whole host of problems. They have, let's see, dilation of heart, so there's a whole bunch of, this, unfortunately I can't read it, but down here it has heart failure, kidney failure, paralysis, pneumonia, and
failure spleen.
What was that?
Failure of spleen.
Failure of spleen. So this one nucleotide is affecting a whole bunch of things, going through a whole different series of stuff. Basically, we know what happens. If you're a sickle cell homozygous, the cells will sickle, and when they sickle, they die and they get stuck in the capillaries. And when they get stuck in the capillaries, that causes circulation to drop out in that region. So you have a whole bunch of problems which are associated with that. And so that's the kind of thing. You have one gene which has one very simple action, but it can have manifest effects all through the body. And that's what pleiotropy is. And then finally I have, in reference to Landy, I have a couple of slides on brain body size allometry. And here we have body weight and brain weight. And what we see is that there's a very nice line here. These are the reptiles and cold-blooded animals. Bony fish and reptiles right through here. Another line for mammals and birds. And then yet another line for primates. And humans are up there. So what that's telling us is that this line, that's essentially, if we take the Chevreul conjecture, that is the, that's a joke. But anyway, if we assume that the phenotypic covariance matrix parallels the additive genetic covariance matrix, this line tells us that this is, that all of the evolution along that line could take place by selection on either trait. And very likely it's selection on body weight. So in other words, to the extent that these are the same line, that is essentially correlated response to selection for body weight. On the other hand, we can see that there's very clearly 3 different lines here. And that they have 3 different intercepts. If we extend these back here, they all have 3 different intercepts. And those difference in intercepts mean selection directly on the brain. That is different selection acting on the brain than the body size. And so what that's telling us is that, in fact, yeah, we have big brains not because we're big. We have big brains because selection was actually acting on primate brain size. This just shows the same thing again. Here's the higher vertebrates. Here's humans. Here's porpoises. So porpoises are right up there as well. Australopithecines down here. You can see we have this move straight up from there. And again, that suggests that basically there's been selection for humans to be smarter. And I think that, yeah, that's all I really have to say about this. And I can go on at this point and talk about other things. I've talked for a while, and stuff I was imagining that I would start talking about tomorrow. But if anybody has any questions, I would be happy to field them. Now, what kind of, anybody? Okay, I will say that we can use any sorts of traits we want to. I didn't think I'd have time for this, but I can actually pull that up. That would be kind of fun. Let's see.
I know some guys around here are interested in modularity, so...
In what?
Modularity.
Modularity. Ah. I don't know if I have a whole lot to say about that.
They're too quiet, so I'm just trying to...
Yeah, no, I was going to say that I actually, something I wasn't sure I would have time for, is to talk about another kind of stuff. And this is in this talk, and I was intending to put it together. We don't need to talk about that stuff. But modularity is an interesting point because modularity is an interesting thing to talk about a little bit, because one of the ideas is that it would be very hard to evolve anything if everything interacted with everything. So if you think about this, you know, if you remember cars, when we first started with cars, They had wind-up windows. Well, when I was a kid, they had wind-up windows. And today, they have a little button on the side with a motor in it. Now think about how they did that. They didn't start over again and redesign the car. What they did was they took out that hand crank mechanism and put in a motor that was the same size. It didn't affect the functioning of the car. And that's the sort of thing that we're talking about with modularity. When we take this unit and by just simply, as we get better car line raising mechanisms, we can simply swap out the old one and put in a new one. And that's what we mean by modularity. We can make small changes where different areas act more or less independently of each other. It actually has a lot of implications. If you think, we haven't really talked about epistasis much, but if you imagine that there's several models of epistasis. One is where you can have a very rough world. If you imagine if you took every time you had a new genotype, you made one change, everything changed. So if the fitness was a random function of genotype, that's called a very rough landscape. And what happens is that if two individuals mate with each other that have very similar phenotypes, or very similar genotypes, their offspring, their phenotype can be anything. And it turns out that you don't get, that when you do that, selection acts very strongly to limit sex, to prevent sex from happening. Because basically, it's a wild crapshoot. Every time you mate, you could produce, you could produce anything. On the other hand, that's not the way the world works. That is, when two humans have sex, they produce a baby which looks kind of like both of them, and is somewhere in the middle. And it turns out that the only way that sex actually works is if you have substitutability. That is, in other words, you don't have to have, it's that two similar genotypes produce similar phenotypes. And that's generally what we find. And one of the ways to do that is to have, in order for that to happen, you have to have your elements be independent of each other. And we'll see this happening in a number of different places. Now, I don't think so much about pleiotropy, and Jim's probably the better one to talk about modularity within organisms. I don't know if you can talk about that. But genetically, one of the things that we'll find happening, and again, I was going to talk about that tomorrow, is that we'll talk about genetic drift. And one of the things that we'll see is that we can have wild epistasis, very heavily interacting epistatic genetic effects. And that as long as we have relatively small population sizes, a lot of those epistatic effects will go away. And we do get modularity in the sense that genes act as if there were no genetic interactions. And sex works. So in a wildly epistatic world, there should be no sex. But in fact, we do have sex. And it really suggests that it's because gene frequency, that we don't have that wild epistasis. That is the distinction between physiological and statistical epistasis, which I haven't gotten into at all. What I wanted to mention very briefly is that one of the things that I work on is multi-level selection. And this is a really interesting thing. Contextual analysis is something I've worked on quite a bit. And in this case, we can imagine a whole bunch of traits. And where it's really easy to imagine brain, body size, and allometry, in this case, what I've done is I've said we can have a bunch of different traits, like brain and body size. But we can also have group mean traits. And so what we're doing is we're saying we can just, when we deal with these multiple traits, we can shove all kinds of crazy things into that P matrix. And for example, we could have the mean milk yield of your cows, the mean butter fat of your cows. We can also have what I call contextual traits, which is the population density, or the running speed of the fastest individual, or something like that. We can throw all of those in. And it's a very interesting thing, because now an individual has a trait, which is the population size they experience. And so everybody who's a member of the same group will have the same phenotype for population size. And we can just simply throw that in, and we can throw in exactly the algebra we've already talked about. And this just shows you the range of different sorts of traits we can use. When we do this, as I say, we assign, this talk is about individuality. And in that I point out that there's only one level at which we assign individuals, and that is, we assign fitness, and that is the level of the organism or the individual. But even so, they have these funny traits, which we can add in there, which is the population size they experience. When we do this, we can then say if we see a significant fitness effect on a group trait, well, that's group selection. Because selection is acting on a composite trait, which is shared by everybody in the population. This was so wild when I started it out that I actually set out to prove that it wouldn't work. And so I actually set up a situation where I said, let's imagine that we have a single trait, Z, and we have for the ith individual in the jth population, sorry, the jth individual in the ith population. And then for each population, we're just going to use the mean of that population. So Zi bar, blue Z's, are the group means. And when we put those two together, you can see that this is a very simple trait, but it is a multivariate model where we have selection acting at the individual level, we have a trait at the individual level and a trait at the group level. And these are two traits which are correlated with each other. And so then I applied different fitnesses to this model, again, purely theoretically. And I said, let's imagine we group individuals together. And we can imagine that we have a fitness which is assigned strictly at the group level, strictly on that group level phenotype. And so all the members of this group have a low fitness regardless of their phenotype. All the members of this group have an intermediate fitness regardless of their phenotype. And all the members of this group have a high fitness regardless of their phenotype. And so, for example, this individual has a high fitness in this population and a low fitness in this population. We can again see that fitness is a function of group membership. We can see that selection is on this group level trait. In the hard selection, it turns out some groups are big, some groups are small, but the fitness of an individual is strictly a function of what group they belong to. And soft selection is where every group has the same fitness. They put out exactly the same number of individuals, but within each group, the big individuals win. So this individual has a high fitness in this group, but if it migrates over here, it's a loser. Yeah, so again, you can see that here we have selection acting only on the group, on Zi, the group trait. Here it's acting only on the individual trait, and here it's acting on both. And it turns out that this is exactly what we find when we do contextual analysis. When we do this multiple regression technique, throwing in group traits as if they were individual traits, what we find is that group selection comes out with all the selection on the group level and not on the individual trait. But again, we have to do the partial regression because, in fact, BW's red Z will be non-zero. Hard selection, we get the opposite result. Selection that we have hard selection, a model where the fitness of an individual is strictly a function of its own phenotype. We see selection acting is acting strictly at the individual level, and there's no group selection. And with soft selection, we see that group and individual selection, are acting exactly in opposition to each other. And I just kind of want to mention this because I want you to see the breadth of which this, while this, again, it seems like a fairly simplistic movement into linear algebra. In fact, it's very powerful. And we can incorporate not just different traits in different individuals, we can also start incorporating neighborhood effects. Yep. And then this is also shows the importance why we have to look at the gradient. why we have to look at partial regressions. Because in this case, if we look at group selection, the partial regression shows group selection acting and no organismal selection. But if we were to only do symbol regressions, which is the easy thing to do, what we would discover is that selection of the regression of relative fitness on individual phenotype is equal to this, which is emphatically non-zero. So in fact, we see that, again, the multiple regression approach ends up being a much more effective way of working with this. Hard selection does the same thing. Okay, so anyway, that's, I think, that's really where I need to stop if we're going to talk about just multivariate selection. And I'm willing to go on, but that starts moving into population structure. I don't know if you guys want to learn about FST today or not. Questions? And I can. I mean, if we have time, I'm happy to keep going. So any questions? I really am happy to answer them. I'm happy to go on. I don't know what you want to do at this point. It's about 11 o'clock. Yeah.
Back to this issue of modularity. If you have some kind of selection on the phenotype of modules being formed and being fixed over time, can you imagine that there would be some kind of selection on the that could be measured on the epistatic effects and pleiotropic effects? Can you study that?
You know, it's really very interesting because selection, when you select on any system with epistasis, what you find is that there's a conversion of, you know, one of the things which I'll talk about tomorrow is that there's a conversion by genetic drift of epistatic effects into additive effects. And so, essentially, what you expect within a population is for epistasis to go away. Same thing happens with selection. Selection, if you really think about it, what selection does is it causes some families to be overrepresented relative to other families. So the effect of selection is effectively to lower the population size. And it does its own conversion. It also causes conversion. This is a really interesting thing because the classic thing we think about in normal systems without genetic interactions, what we expect is that selection should use up genetic variation. But that's not what we see when there's epistasis. It actually increases the additive genetic variance. And so as a result, what that will do is it essentially makes the epistatic interactions go away, and as a consequence, makes the leftover genetics more and more additive. And so when you go into a population that's been small or under selection, you expect to see very little epistasis as a variance component. And I suspect the same thing would happen with modularity, that essentially those individuals that have negative interactions in area between modules are going to lose. And so you'll essentially select for individuals which have independent sex. And that's where I was trying to bring in sex, is in fact, sex is almost universal. And sex does require that the offspring be predictable. If the offspring were not predictable, it wouldn't be. And so that's sort of it appears to be a natural outcome of evolution, is that some level of modularity occurs. And you know, most of the time we expect to see gene interaction, we expect to see it between populations. Do you have anything you want to add to that?
Talk some later about the modularity.
Okay, because I'm kind of talking off the top of my head a little bit on that. So, okay. So what do you want to do? You want to stop here and
make them read.
Okay, make them read. And I'll be happy to talk to anybody who has questions about any of this. One of the things I've definitely not talked about is how you measure this stuff.